Hi students, myself Satish Kumar Kunidala, Assistant Professor, Department of Pharmaceutical Sciences, Vignan's Foundation for Science, Technology and Research, Guntur. In this video, we are going to discuss about the cholesterol, and that we are going to discuss about biological role of the cholesterol, biosynthesis of cholesterol, degradation process of the cholesterol, and finally we are going to discuss about the disorders associated with the lipid metabolism. Okay, let us see one by one. The cholesterol is found excessively in animals, hence it is also called as animal sterol. So cholesterol is a sterol molecule. So it is excessively found in the animals, that's why it is called as the animal sterol. The total body content of cholesterol in an adult man of weight of having the weight of 70 kg is about 140 grams. So the total cholesterol present in the a human weigh about 70 kgs. So 70 kg human having the total body cholesterol is about 150 gram. 140 grams. Okay, and that is about 2 grams per kg. So 2 grams per kg body weight. And cholesterol is amphipathic in nature since it is possessing the both hydrophilic and hydrophobic regions in the structure. So the chemical structure of cholesterol having the both hydrophilic and hydrophobic regions are functional groups. That's why it is called as the amphipathic molecule. Okay, amphipathic molecule. Amphipathic molecule. And coming to the functions of cholesterol, so this one is essential for the life as it performs a number of important biological functions, like it is a structural component of the cell membrane. So in the cell membranes, in cell membrane, it is one of the structural component, and this cholesterol is a precursor for the synthesis of other steroids like steroid hormones, vitamin D, bile acids, and is also essential for ingredient in the structure of lipoproteins, and it is also useful for the fatty acids transport in the liver in the form of cholesterol esters. So then they are subjected to oxidation. So cholesterol is required for the transport of fatty acids in the liver for their oxidation or degradation purpose. So that is a biological functions of the cholesterol. And coming to the biosynthesis of cholesterol, how the cholesterol is synthesized in the biological system, you can describe here. So about 1 gram of cholesterol is synthesized per day in the adult. So for each day, 1 gram of cholesterol is synthesized in the adults. Almost all the tissues of the body participate in the cholesterol biosynthesis. The largest contribution is made by the liver. So the 50% of the cholesterol is synthesized in the liver. That means for every day 1 gram is produced. Out of that 1 gram, 50%, that means 0.5 grams is produced from the liver, and 15% is produced from the skin. So skin produces 15% of the cholesterol. Okay, and remaining adrenal cortex, reproductive tissue. So these all are will provide the remaining, will produce the remaining cholesterol. And the enzymes involved in the cholesterol biosynthesis are found in the cytosol and microsomal fractions of the cell. So the enzymes which are involved in the cholesterol biosynthesis are present in the cytosol. Cytosol means, so inside the cell the fluid is there, so that is called as the cytosol. So the enzymes present in the cytosol as well as the microsomal fractions of the cell. And acetate of acetyl CoA provides all the carbon atoms in the cholesterol. So we know that there is a cholesterol structure. So the cholesterol structure may be like this. This is the structure of cholesterol. So this is the structure of cholesterol. So the carbon atoms which are present in the cholesterol structure. So these are the carbon atoms that are present in the cholesterol structure. So these carbon atoms are provided from the estate of acetyl CoA. So the estate of acetyl CoA. Estate of acetyl CoA will produce all the carbon atoms in the cholesterol. The reducing covalents are supplied by the NADPH, and ATP will provide the energy for the biosynthesis of this cholesterol. So there are three prerequisites for the biosynthesis. First one is the acetyl CoA. It will provide the acetyl group of this acetyl CoA. Acetyl group of acetyl CoA will provide carbons of cholesterol, and NADPH. NADPH will produce or gives the reducing equivalents or supplies the reducing equivalents. Supplies reducing equivalents. And ATP is utilized as the energy in the biosynthesis of this cholesterol. Okay. And for the production of 1 mole of cholesterol, so for example, if you want to produce 1 mole of cholesterol, so for that reason 18 moles of acetyl CoA is required and 36 moles of NADPH, 36 moles of ATP and 16 moles of NADPH are required. So these are the requirement for the biosynthesis of 1 mole of cholesterol. For producing 1 mole of cholesterol, we require 18 moles of acetyl CoA, 16 moles of NADPH and 36 moles of ATP. Okay, and by administering acetate with 14 carbon isotope label either on the methyl group or carboxyl group, then the origin of carbon atoms in the entire molecule of cholesterol has been established. So that means the 14 carbon isotope, so labeled either on the methyl group of carboxyl group or methyl group or carboxyl group, the origin of the carbon atoms in the entire molecule. So that is so when you number these, and coming to the synthesis of cholesterol, so that includes 5 steps. So they are nothing but synthesis of HMG CoA, formation of mevalonate and production of isoprenoid units, synthesis of squalene and conversion of squalene to the cholesterol. So these are the 5 steps involved in the synthesis of cholesterol. So there are 5 steps involved in the synthesis of or biosynthesis of cholesterol. Here we can see the structure of cholesterol. It is having the hydroxyl group at this position and also having the substitutions like this. So this is the structure of cholesterol. Okay. So here we can see the structure of cholesterol. So it is numbered like this. So starts from here. So it starts from here 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 like that. It will start the numbering. And administration of acetate. So the 14 carbon labeled acetate. The 14 carbon labeled acetate. So that is labeled on either methyl group or so this is the carboxyl group. So this one is the methyl group. So this is the methyl group and this one is the carboxyl group. So when you label either on the methyl group or carboxyl group of the 14 carbon acetate, so then the total carbons which are present in the cholesterol is established. So here the methyl group will provide the carbons which are numbered like here the first carbon, third carbon, fifth carbon, seventh carbon, ninth carbon, 13th carbon, 15th carbon. So like that, so all carbons which are numbered here, so that carbons are provided from the methyl group and remaining carbons, so that is second carbon, fourth carbon, sixth carbon, Okay, so all these carbons are provided from the carboxyl group. So these are the estate moiety which can produce the carbons that are construct the cholesterol molecule. So the acetyl group of acetyl CoA. So the acetyl group of acetyl CoA, which is having the 14 carbons, so that can establish the origin of cholesterol molecule biosynthesis. So in that also the methyl group of acetate will produce or will contribute the carbons that are numbered like first carbon, third carbon, fifth carbon, seventh carbon, eighth carbon, 13th, 15th, 17th, 18th, 19th, 21, 22nd, 24, 26, 27 carbons are produced from the methyl group of acetate, and remaining carbons like second carbon, fourth carbon and so on are produced from the carboxyl group of the acetate. Okay. And here we can see the brief review or brief flow of this cholesterol biosynthesis. So it starts from the acetyl CoA. The cholesterol biosynthesis will start from the acetyl CoA. So that is a 2 carbons of acetyl CoA. The acetyl CoA consisting of 2 carbons, and it will be converted to HMG CoA. That is the first step involved in the biosynthesis of cholesterol. So production of HMG CoA. So it is having the 6 carbons. Then it will be converted to malonate. That is the second step. So the malonate will consisting of 6 carbons. So first starts with these 2 carbons, then it is a 6 carbon, and malonate also 6 carbons, and isoprenoid units, that is a 5 carbon building blocks, and 6 units of these isoprenoid units condense to form the squalane. So 5 into 6, 30 carbons will form present in this squalane. Then it will be converted to lansosterol. So that is also 30 carbon molecule, and this lansosterol is converted to cholesterol. So this is the major steps involved in the biosynthesis of cholesterol. Okay. So here we can see the structural modifications of these acetyl CoA to the cholesterol. So these are the remaining steps involved in this biosynthesis of cholesterol, and we can see each and every step in detail. So this is the first step in the biosynthesis of cholesterol, that is nothing but synthesis of beta hydroxy beta methyl glutaryl CoA, that is nothing but the HMG CoA. So the production of HMG CoA is the first step involved in the biosynthesis of cholesterol. The HMG CoA is nothing but beta hydroxy beta methyl glutaryl CoA. Okay, and for that we require acetyl CoA. So the 2 molecules of acetyl CoA condense to form the acetyl CoA. So here first the 2 molecules of acetyl CoA condense to form the acetyl CoA by the action of enzyme, that is a thiolase. So the thiolase will catalyze the condensation of 2 molecules of acetyl CoA to form the acetyl CoA. Okay, and another molecule of acetyl CoA is then added to produce the HMG CoA. So again there is a acetyl CoA. So this is the acetyl CoA. Again one acetyl CoA is involved, or that will be added to this acetyl CoA to produce the beta hydroxy beta methyl glutaryl CoA, that is a HMG CoA. And this reaction, or the conversion of acetyl CoA to HMG CoA is catalyzed by the HMG CoA synthase. This is the enzyme, so that will catalyze the conversion of acetyl CoA to beta hydroxy beta methyl glutaryl CoA or HMG CoA. So I will repeat it once again, the formation of production of HMG CoA. So it will start from the 2 molecules of acetyl CoA. The 2 molecules of acetyl CoA will condense by the action of thiolase to form the acetyl CoA, and this acetyl CoA by the action of HMG CoA and will be added to another acetyl CoA molecule, then it will form the beta hydroxy beta methyl glutaryl CoA. That is a HMG-CoA. And these reactions are similar to that of the ketone body synthesis. However, the two pathways are different since ketone bodies are produced in the mitochondria, while this biosynthesis of cholesterol is happened in the cytosol. So that is happened in the ketone body synthesis is happened in the mitochondria, but cholesterol synthesis occurred in the cytosol. Okay, so both are similar. So appearance is similar, but the location of these pathways are different. So that is occurred in the mitochondria, and this is occurred in the cytosol of the cell. Okay, and further two isoenzymes of HMG-CoA synthesis are known. Sorry, the two coenzymes of HMG-CoA synthesis are known. So the cytosomal HMG is involved in the cholesterol synthesis, whereas the mitochondrial HMG-CoA synthase is involved in the ketone body formation. So here, so when seen this HMG-CoA synthase, so this is the cytosol HMG-CoA synthase. There are two types of HMG-CoA synthases. One is the cytosol HMG-CoA synthase, and another one is a mitochondrial HMG-CoA synthase. So cytosomal or cytosol HMG-CoA synthase is involved in the biosynthesis of cholesterol, and mitochondrial HMG-CoA synthase is involved in the production of ketone bodies in the mitochondria. Okay. And coming to the second step of cholesterol biosynthesis, that is nothing but formation of mevalonate. So in first step, we seen that for production of HMG-CoA, that is nothing but the beta hydroxy, beta methyl glutaryl CoA. So this is the structure of beta hydroxy. So here it is so this is the beta carbon. So beta hydroxy, beta methyl glutaryl CoA. So this is these carbons are called as glutaryl group. So there is a CoA is there. So that's why it is called as beta hydroxy, beta methyl glutaryl CoA, and this molecule is converted to mevalonate by the action of HMG-CoA reductase. So the HMG-CoA reductase is the enzyme that will convert the HMG-CoA to mevalonate. So this is a rate limiting step in the cholesterol biosynthesis. Why? Because the HMG-CoA reductase can be inhibited by some of the compounds. For that reason, it is called as the rate limiting step of cholesterol biosynthesis. So this enzyme is present in the endoplasmic reticulum. So that catalyzes the reduction of HMG-CoA to mevalonate, and the production of reducing reducing equivalents are supplied by the NADPH. So for this reaction, the NADPHs are utilized to reduction process of this HMG-CoA to mevalonate. The HMG-CoA reductase will reduce the beta hydroxy, beta methyl glutaryl CoA or HMG-CoA to mevalonate. For this reduction purpose or for a reduction process, so there is a two NADPHs are utilized, so to convert this HMG-CoA to mevalonate. So this HMG-CoA reductase is produced in the endoplasmic reticulum. Come to the third step of cholesterol biosynthesis is nothing but production of isoprenoid units. In the second step, we seen that production of mevalonate, and this mevalonate is converted to 5-phosphomevalonate. 5-phosphomevalonate by the action of mevalonate kinase. For this purpose, there is a 1 ATP is utilized. Okay, so for conversion of mevalonate to 5-phosphomevalonate, so there are 3 steps involved in the production of isoprenoid units. In that, first step is mevalonate is converted to 5-phosphomevalonate, then it is converted to 5-pyrophosphomevalonate, and it is converted to 3-5-pyrophosphomevalonate. So that is the steps involved in the conversion of mevalonate to isoprenoid units. So we'll see this in detail. First, the mevalonate is converted into 5-phosphomevalonate. So here it is a hydroxyl group is present in this 5th carbon, and this will be converted to phospho, the hydrogen is removed and phospho group is attached at this position. So that's why it is called as the 5-phosphomalonate. So this reaction is catalyzed by the malonate kinase. This is the malonate kinase enzyme. So for this purpose, there is a ATP is required. ATP is utilized. And this 5-phosphomalonate is converted to 5-pyrophosphomalonate. 5-pyrophosphomalonate. That means again other phosphate group is attached to this. So at this position, again one phosphate group is attached. So this reaction is catalyzed by the pyrophosphomalonate kinase. Pyrophosphomalonate kinase. That is an enzyme. So for this reaction also 1 ATP is required. 1 ATP is required. So first the malonate is converted into 5-phosphomalonate. Then it is converted to 5-pyrophosphomalonate. That means phosphate group is attached in each step. Okay. Next, the 5-pyrophosphomalonate is converted to 3-5-pyrophosphomalonate. So this reaction is catalyzed by the enzyme kinase. This is the enzyme kinase. So this kinase will catalyze the conversion of 5-pyrophosphomalonate to 3-5-pyrophosphomalonate. That means the phosphate group is attached at the third carbon also. So here it is. So the phosphate group is attached here, the third carbon. So already two phosphate groups are attached at the fifth carbon. That's why it is called as the 5-pyrophosphomalonate. And again the phosphate group is attached at the third carbon. That's why it is called as the 3-5-pyrophosphomalonate. Then this 3-5-pyrophosphomalonate is converted into isopentenyl pyrophosphate. Isopentenyl pyrophosphate. So this will be catalyzed by the enzyme that is a pyrophosphomalonate dehydrogenase. Pyrophosphomalonate dehydrogenase. So this enzyme will catalyze the conversion of 3-phosphate 5-pyrophosphomalonate to isopentenyl pyrophosphate. Okay. And again this isopentenyl pyrophosphate will be converted to dimethyl allyl pyrophosphate. So this molecule is called as dimethyl allyl pyrophosphate. So the conversion of isopentenyl pyrophosphate to the dimethyl allyl pyrophosphate is catalyzed by the isopentenyl pyrophosphate isomerase. So this is the enzyme. So this enzyme will catalyze the conversion of isopentenyl pyrophosphate to dimethyl allyl pyrophosphate. The enzyme name is nothing but isopentenyl pyrophosphate isomerase. So these two, so this molecule, there is a isopentenyl pyrophosphate and dimethyl allyl pyrophosphate are two molecules are called as the isoprenoid units. Both are called as the 5-carbon isoprenoid units. We know that isoprenoid units consisting of 5 carbons. So here we can see both isopentenyl pyrophosphate having the 5 carbon units, and also here it is dimethyl allyl pyrophosphate also called as the 5 carbon units. But malonate consisting of 6 carbon units. The 6 carbon unit will be converted into 2 isoprenoid units that consisting of 5 carbons in their structure. Okay, that is the third step of cholesterol synthesis. And coming to the fourth step of the biosynthesis of cholesterol, that is nothing but synthesis of squalane. In third step, we seen that production of 2 isoprenoid units, that is nothing but isopentenyl pyrophosphate and dimethyl allyl pyrophosphate. So these 2 isoprenoid units condense to produce the gernyl pyrophosphate, that is a 10 carbon molecule. So here the isopentenyl pyrophosphate and dimethyl allyl pyrophosphate will condense to produce the gernyl pyrophosphate. So this reaction is catalyzed by the cis prenyl transferase, that is an enzyme. So this enzyme will convert the or condense the two isoprenyl units, that is nothing but isopentenyl pyrophosphate and dimethylallyl pyrophosphate. These are the two isoprenyl units, each consisting of high carbon in their chemical structure, and they are condensed to form the gernyl pyrophosphate, that is a 10 carbon molecule. So this reaction is catalyzed by the cis prenyl transferase, and again this gernyl pyrophosphate, that is a 10 carbon molecule, again condensed with the other isopentenyl pyrophosphate molecule. There it can form the farnesyl pyrophosphate. So this reaction also catalyzed by the cis prenyl transferase. So that is an enzyme that can condenses the prenoid units to form the farnesyl pyrophosphate, and then the two molecules of farnesyl pyrophosphate, so two molecules of farnesyl pyrophosphate reduced to form the squalane, so the two units of farnesyl phosphate units are get reduced, thereby it can form the squalane. So that is a 30 carbon molecule. This reaction is catalyzed by the squalane synthase. So that is an enzyme which will catalyze the reduction of two farnesyl pyrophosphate units to produce the squalane. So here also there is a one NADPH is utilized. NADPH is utilized for the reduction of two farnesyl pyrophosphate molecules to the squalane. Okay. So the reduction equivalents are produced from the NADPH. Okay. So wherever the reduction takes place, there the NADPH is utilized. And coming to the fifth step, that is the last step of cholesterol biosynthesis, is nothing but conversion of squalane to cholesterol. So in fourth step, we've seen that production of the squalane from the isoprenoid units, and that produces squalane is converted to cholesterol in this final step. So the squalane undergoes hydroxylation and cyclization by utilizing the oxygen as well as the NADPH. So thereby it will be converted into lansosterol. So this reaction is catalyzed by the epoxidase hydroxylase cyclase. That is an enzyme that will convert the squalane to lansosterol by utilization of oxygen and NADPH. Okay. So this formation of cholesterol, from this lansosterol is multi-step process. That means so many steps are involved during the conversion of lansosterol to the cholesterol. So about 19 steps or 19 enzymatic reactions are involved during the conversion of lansosterol to cholesterol. So that is nothing but a reduction of the carbon atom from 30 to 27. We know that the lansosterol and squalanes are 30 carbon compounds. These two are the lansosterol and squalanes are the 30 carbon compounds. So this 30 carbon compounds will be converted into 27 carbon. Cholesterol is a 27 carbon molecule. So there is a conversion of 30 carbon molecule to 27 carbon molecule, and removal of two methyl groups from the C14. That is from the fourth carbon atom, there is a removal of two methyl groups. So there is a removal of two methyl groups from the fourth carbon atom and one methyl group from the, so there is a one methyl group from the 14 carbon, and there is a shift of double bond from the 8th carbon to 5th carbon. From 8th carbon to 5th carbon, there is a one double bond is shifted, and reduction in the double bond present between the 24th carbon and 25th carbon. So there is a reduction of, so when you see in this structure, so these operations, these transformations are done during the 19 enzymatic reactions. During these 19 enzymatic reactions, so these are the things which are going to be done to convert the lansosterol to cholesterol. Okay, and the enzyme, that is a 19 enzymes are involved in the conversion of lansosterol to cholesterol, and they are associated with the endoplasmic reticulum. So the all enzymes which are involved in the conversion of lansosterol to cholesterol are associated to the endoplasmic reticulum, and at the last stage we can see the during this conversion of lansoprazole to the cholesterol, there are some intermediates like 14 desmethyl lansoprazole, gymosterol, cholesterol dienol, and decimosterol. These are the some intermediates that are formed during the conversion of lansoprazole to the cholesterol. So the intermediates are nothing but 14 desmethyl lansoprazole, gymosterol, cholesterol and demosterol. These are the intermediates. And at the last, we can see the formation of 7 dehydrocholesterol. So that will be converted to cholesterol by the reduction process. So at the end of this conversion of the lansoprazole, so there is a formation of 7 dehydrocholesterol, and that will be converted to cholesterol by the reduction process. So this is the process involved in the biosynthesis of cholesterol. In cytosol of this cell. And coming to the degradation of cholesterol, so the steroid molecule, so the steroid molecule is nothing but, this is the steroid molecule which is present in the cholesterol. This is a steroid molecule, so it is present in the cholesterol. So this nucleus is not metabolized in the humans. So the nucleus, steroid nucleus which is present in the cholesterol structure is not metabolized in the humans. So 50% of cholesterol is converted to bile acids and excreted in the feces. So 50% which is produced in the humans or which is present in the human, that will be converted into bile acids and that will be excreted through the feces. And remaining cholesterol will serve as the precursor for the synthesis of steroid hormones like sex hormones, vitamin D, coprostanol and cholestanol. So these are the some products which are produced by the cholesterol or from the cholesterol. So the latter two are fecal steroids and these are produced from the cholesterol. So the two, the coprostanol and cholestanol, these two are the fecal steroids. So they are produced from the cholesterol. So here we can see the biosynthesis of bile acids from the cholesterol. So we know that cholesterol will act as the precursor for the synthesis of bile acids, steroids, some other coprostanol and cholestanol. So these are the, and vitamin D also produced from the cholesterol. So we can see the biosynthesis of these compounds. So first we will see about the synthesis of bile acids from the cholesterol. So bile acids possess 24 carbon atoms, but cholesterol consisting of 27 carbon atoms, and bile acids possess 24 carbon atoms, and 2 or 3 hydroxyl groups are present in the structure that are present are attached to the steroid nucleus, and there is a side chain ending with the carboxyl group, but the side chain is ended with the methyl groups in the cholesterol, but here the side chain ends with the carboxyl group in the bile acids. Okay, and these bile acids are amphipathic in nature. Amphipathic means both consisting of both polar and non-polar groups. So cholesterol is also a amphipathic in nature. Okay, they serve as emulsifying agents in the intestine and actively participate in the digestion of or absorption of the lipids. Okay, so it is a role of bile acids. They are acting as the emulsifying agents in the intestine, and thereby they can participate in the digestion and absorption of the lipids. And the synthesis of primary bile acids takes place in the liver and involves a series of reactions. So let me see that series of reactions that are involved in the synthesis of bile acids that are occurred in the liver. So here are the some series of reactions that can be involved in the synthesis of bile acids in the liver. The first step is nothing but conversion of cholesterol to 7-hydroxy-cholesterol. So this is the first reaction involved in the biosynthesis of bile acids from the cholesterol. So for that reason, there is a first step, that is a conversion of cholesterol to 7-hydroxy-cholesterol. So this reaction is catalyzed by the 7-alpha hydroxylase. So this 7-alpha hydroxylase is inhibited by. So this is inhibited by the bile acids. So that means the produced bile acid will inhibit the 7-alpha hydroxylase. Thereby, this reaction is the rate limiting step of the bile acid synthesis. If the bile acids are more, then it will prevent the or it will inhibit this enzyme. Thereby, there is no production of bile acids occurred in the liver. So that's why it is considered as the rate limiting step of bile acids synthesis. Okay, and from this 7-hydroxy-cholesterol, there is a production of deoxycholic acid, and another one is a lithocholic acid. Lithocholic acid. There are two types of acids are produced from the 7-hydroxy-cholesterol. So in that first one, there is a production of cholic acid and chenodeoxycholic acids are formed. These two are considered as the primary bile acids. So these two are produced in larger amounts. So for the production of these cholic acid and chenodeoxycholic acid, several steps takes place between these reactions. Several reactions takes place. That means for conversion of 7-hydroxy-cholesterol to either cholic acid or chenodeoxycholic acid, there are several reactions or several steps will take place in the liver, and finally the cholic acid and chenodeoxycholic acids are produced. So these two are produced in large amounts. Okay. On conjugation with glycine or taurin, conjugated bile acids like glycocholic acid or taurocholic acids are produced, and which are more efficient in their function as surfactants. So there is a two acids are produced when the conjugation of glycine or taurin to the cholic acid. So that they are nothing but glycocholic acid or taurocholic acids. These two are more powerful in their action as the surfactants. Okay. In case of chenodeoxycholic acid, that will be able to form the taro-glycochenodeoxycholic acid. That is also a powerful surfactant. And in the bile, the conjugated bile acid means glycocholic acid or taurocholic acid, or it is also, or another one is a taro-glycochenodeoxycholic acid. These are exits as sodium and potassium salts, which are known as the bile acids. So when they are combined with the sodium or potassium to form as or to appear as the salts in the bile acids, so appeared as the salts in the bile. So this is about the biosynthesis of bile acids from the cholesterol. So I will repeat once again. So there is a conversion of cholesterol. First step is nothing but conversion of cholesterol to 7-hydroxy-cholesterol or cholesterol. That is this reaction is catalyzed by the 7-alpha-hydroxylase. So this is the rate limiting step. Why? Because this enzyme is inhibited by the bile acids, and then the 7-hydroxy-cholesterol is converted to cholic acid and chenodeoxycholic acid by different or several steps or several reactions, and these Cholic acid and chenodeoxycholic acids are considered as the primary bile acids. They are produced in large amounts in the bile, and these primary bile acids on conjugation with the glycine and taurin, they can produce the glycocholic acid or taurocholic acid. So these are considered as the conjugated bile acids. So they are very powerful in their function as the surfactants, and these conjugated acids will combine with the sodium or potassium. They will appear as the salts in bile. Okay. Next, coming to the biosynthesis of steroid hormones from the cholesterol. So how the steroid hormones are produced from the cholesterol. So the cholesterol is a precursor for the synthesis of all 5 classes of steroid hormones. So the 5 classes of steroid hormones are nothing but progestins. The example for these progestins are progesterone, glucocorticoids, example is cortisol, mineralocorticoid, that is a example is aldosterone, androgens, example is testosterone, estrogens, example is estradiol. Okay, so these are the 5 classes of steroid hormones that are produced from the cholesterol. So here is the process involved in the steps involved in the synthesis of steroid hormones from the cholesterol. So the cholesterol will be converted into pregnenolone, then it is converted to progesterone, then it is converted to cortisol, androsterone and estradiol, like this. So these are the some steps involved in the conversion of cholesterol to steroid hormones. Next we will discuss about the synthesis of vitamin from the cholesterol. So during the conversion of lansosterol to cholesterol, there is a formation of 7-dehydrocholesterol is formed. So it is an intermediate of cholesterol synthesis from lansosterol. Okay, and so this will be converted to calciferol. So cholecalciferol. So the 7-dehydrocholesterol. The 7-dehydrocholesterol, that is an intermediate of cholesterol from conversion of during the conversion from lansosterol to cholesterol, there is a last end product is nothing but 7-dehydrocholesterol. So this will be converted to cholecalciferol. So cholecalciferol is also called as vitamin D3. Vitamin D3 by ultraviolet rays in the skin. So there is a last or 5th step of cholesterol synthesis. So that is nothing but conversion of lansosterol to cholesterol. So before converting this lansosterol to cholesterol, there is a formation of 7-dehydrocholesterol. 7-dehydrocholesterol will take place. So this 7-dehydrocholesterol by reduction it will be converted to cholesterol. By reduction it will be converted to cholesterol. So before converting the 7-dehydrocholesterol to cholesterol, the 7-dehydrocholesterol is converted to cholecalciferol. So there is a vitamin D3. Vitamin D3. So this reaction is operated or catalyzed by the ultraviolet rays, UV rays. So this will happen in the skin. So when this 7-dehydrocholesterol which is present in the skin is exposed to the UV rays, then it will be converted into vitamin D3. So that's why, so when you expose it to the sunlight, so in the sunlight we are having the UV rays, so that UV rays will convert the 7-dehydrocholesterol to cholecalciferol or vitamin D3. Okay. Here we can see the series of steps involved in the conversion of cholesterol to the cholecalciferol or vitamin D3. So we know that 7-dehydrocholesterol is the intermediate of cholesterol, and that 7-dehydrocholesterol is converted into pre-vitamin D3 by the action of UV light, and this pre-vitamin D3 is converted into cholecalciferol or vitamin D3 by the action of thermal isomerization. thermal isomerization. So there is a formation of cholesterol will takes place from the 7-dehydrocholesterol. Okay. So this cholesterol, this vitamin D3 may be subjected to degradation by the enzyme that so it is thereby it can form the hydroxycholecalciferol or 1,25 dihydroxycholecalciferol. So that is the vitamin D3 also subjected to degradation. So thereby it can able to form the hydroxy vitamin D3 or dihydroxy vitamin D3. So this reaction is takes place by the liver 25 hydroxylase or 1,25 hydroxylase. Okay, so there is a thing which is happened in case of vitamin D3. So these are nothing but the formation of vitamin D. So these two are called as the vitamin D. So this last product 1,25 dihydroxy cholecalciferol is called as the vitamin D. Okay, so this is the production of vitamin D from the cholesterol.